Ah, I see you've been to the farmer's market again. And of course, you would pick the mutant fruit.
What are you talking about? That banana is perfect. No bumps and no bruises.
Well, it's a mutant. Think about it. Haven't you ever wondered why bananas don't have any seeds? None. Isn't that a little weird?
Well, now that you mention it,
It's a mutant. Now, granted, the original mutation happened long ago. We humans decided that we quite liked bananas without seeds, so we continued to grow this mutant variety.
well, the banana is in good company because I can think of another mutant in the room.
I know, the hair. Red hair is also a mutation.
It also explains why you were able to spot the banana so fast. Takes a mutant to know one.
By the end of this week, we'll have covered everything you've ever wanted to know about mutations on
DNA Decoded. So far in this course, we've talked about DNA replication and what happens when everything goes right. Let's quickly recap how DNA is copied. DNA polymerase gets into position at the start location. As it moves along, the DNA polymerase reads the sequence of base pairs using Watson and Crick's rules, A pairs with T and C pairs with G. DNA polymerase finds the corresponding base and attaches it to form a base pair. Let's say DNA polymerase is in the process of building along this segment of DNA. The correct sequence, according to Watson and Crick's rules, would be
Now let's talk about how that DNA is transcribed into messenger RNA. The DNA strands are unzipped to create a transcription bubble. RNA polymerase gets into position at the start location and proceeds to add the Watson and Crick base pairs. The end result is a single strand of mRNA. Now the messenger RNA is read 3 bases at a time by the ribosome. Those triplets are called codons. There are 64 ways to combine the 3 bases to create different proteins. We can translate the mRNA codon into the instructions for building amino acids by looking up which codons map onto which amino acids. If we decode the codons from the messenger RNA for the correct DNA template, here's what we would get. M, L, L, and N are all the amino acids. But what happens when things go wrong? Errors in DNA replication result in mutations. Some are harmless, some are valuable, some are crippling.
mutations are just changes in DNA. They're actually more common than you think. You might think all mutations are bad, but there are plenty of cases where a mutation has no consequence at all. Weird, right? That's biochemistry for you. Let's explain.
There are probably harmless mutations happening inside us right now as our cells divide and new tissues grow. To understand why some mutations make a difference and others do not, we need to introduce 2 terms, synonymous and non-synonymous mutations. Suppose something goes wrong and an A is mistakenly added in to our example of DNA instead of a C. That's a mutation or a mistake in the copying. It looks bad, but let's fast forward through the next steps to see what the consequence will be, which gives us the following RNA sequence. What does this messenger RNA code for? Well, look at that. There was no difference in the amino acid sequence, so there's no difference in the protein created.
More than one triplet can code for the same amino acid. It's like the 2 codes are synonyms for each other. In this case, both messenger RNA sequences produce the same amino acid, leucine, or L.
Even though there was a mutation in a base pair, the mRNA could still build the correct protein. This is a synonymous mutation. It doesn't alter the amino acid sequence. Now let's shift gears and look at another mutation. Here, the error is in C at the 4th position. This means the messenger RNA is, what does the mRNA code for?
Yikes, red alert, Houston, we have a problem. Now we've got a different amino acid sequence.
Sure do. That valine does not belong there. This could mean the protein's function is affected. This is called a non-synonymous mutation because the protein sequence has been altered. Replication mutations are just one kind of mutation. You can find out about other types of mutations in the supplemental materials.
Now let's go back to the mutation example we mentioned at the beginning of this video, the flaming red hair that makes Kate a mutant. Your red hair is caused by a mutation in your MC1R gene. That stands for melanocortin 1 receptor. This protein plays a key role in determining hair and skin color. Here's the cool part. The most common mutation leading to red hair is caused by a single nucleotide change.
Yep, that's right. Just one single nucleotide change can make a big difference. Melanin is the pigment that determines the color of your eyes, skin, and hair. Humans actually produce two types, red and black. The mutation in the MC1R gene in redheads means they produce predominantly red pigment and very little black pigment. There you go, red hair.
As far as mutations go, seedless banana and red hair are pretty harmless, but other types of mutations can have tragic consequences.
Cystic fibrosis is a genetic disease that results from a single gene mutation on chromosome 7. Individuals that suffer from cystic fibrosis have respiratory and digestive disorders. The most common mutation that leads to cystic fibrosis is a deletion mutation. 3 nucleotides are missing, meaning that an entire amino acid is lost. Patients with cystic fibrosis have an impaired protein that results in unusually thick mucus production.
So to recap, sometimes mistakes happen. Mutations are errors, but not all mutations are bad. As we've seen, mistakes are responsible for giving us delicious bananas and for giving Kate her red hair. But sometimes the mistakes have very serious consequences, as we saw with cystic fibrosis patients. This brings us to our next video topic, genetically modified organisms. Stay tuned.